Just a few concluding points only. Vacuoles. Two important points are there regarding vacuoles. Vacuoles are the sac-like structures inside plant cell. They will be big vacuole, very prominent vacuole in plant cell. When you take animal cell, you will have small numerous vacuoles. So these are the terms when you refer to animal cell. Okay, small numerous vacuoles we will find in animal cell. That's the basic difference. Then other than that, the vacuoles contains either waste material or they may store food materials also. Okay, either waste material or they may store useful material also, vacuoles. The membrane around vacuole is the most important point. The membrane around vacuole is called tonoplast. This is the term important one. Write it down. Okay. Vacuoles are surrounded by a single membrane called tonoplast. Other than that, remaining all you know. Then the other points here is they have given cytoplasmic inclusions. In the concluding part of the chapter, they have given this word cytoplasmic inclusions. Means in the starting of the cell structure, I told you two terms, organelle and inclusion. If you remember.
Yes,
cell organelles, yes. Cell organelles are the one which are involved in different functional aspects. They are like living entity of the cell. Organelles, cell organelles like mitochondria, chloroplast, endoplasmic reticulum, and all. Okay. Then inclusion. Yeah, I'm just explaining that point. Inclusion involves non-living one. Non-living means what? The kind of waste materials, the kind of reserve food materials, all those things comes under cytoplasmic inclusions. Inclusions are nothing but the non-material which you will find in the cytoplasm. That's all. Okay. That concludes the whole cell structure and function. When you take the microorganisms, there are various roles microorganisms playing in the whole environment. When you take microorganisms, the microorganisms can be friendly to the environment or they can be enemies to the environment. I'm using the word environment. Environment includes everything. The atmosphere around you in the form of air, the atmosphere which is making part of soil, water, and including plants, animals, humans also. So the living and non-living part, the whole living and non-living part will form the environment, isn't it? So microorganisms might be helpful in some ways and they may be harmful in some ways. So what are different microorganisms and how they are helping the environment, how they are harming the environment, this whole chapter deals with. First we'll start with where the microorganisms will be present. Okay, what the environment the microorganisms are existing. Then we'll get into the classification of microorganisms. Then how each microorganism is useful and harmful. The both sides of the coin we are going to discuss. Organism is nothing but a different concept. Cell is a unit of life, isn't it? Organism is a living entity. Even organisms will also have cells. Even microorganisms also will have cells, isn't it? Cell is the functional unit of life. The life may be in the form of microorganism. So you will find cells in microorganism. There's a difference between cell and microorganism. When you take the microorganisms, the name itself indicates micro. We cannot see these organisms. We cannot see these organisms with naked eye. Through microscope usage only we can see. We are having various types of microscopes. Okay, that includes a simple microscope, compound microscope, electron microscope, and with advancement so many other categories of microscopes came into existence. Now all these microscopes will work at different resolution. Okay, so here I just want to introduce two terms regarding microscopes, so that whenever you use any microscope, you will get an idea of what does that really mean. Now, microscope are the instruments which were discovered far back in 16th century. Actually, the work started itself in 15th century, but the light microscope is the first microscope, first category of microscope, if we can say, in the order of development of microscopes. Then after light microscope, we have an advanced type called electron microscope. In between, we are having a compound microscope. So there's no much difference between light microscope and compound microscope. Then advanced one is electron microscope. Right now we are having fluorescent microscope. Total instrument, what our Anthony van Leeuwenhoek used was exact instrument what we call as microscope. That's a crude example of microscope. But he was the one who used microscope to observe a living cell. Okay. Now coming to the microscopes, these many microscopes I have given. See, what is the difference between all these things? Whenever we talk about microscope, we talk about resolution. Okay, resolution is nothing but how much perfectly or how much in detail you can see an object. That gives the resolution. I'll give in the terms of compound microscope. See, light microscope and compound microscope used to observe the particles or the objects with a diameter of 10 to the power of minus 6 meters, which we call as micron. Okay, 10 to the power of minus 6. Yeah, I'll come to that point, how they take pictures. Okay, so if you take the resolution, is much depth in detail you can observe under a microscope. Even electron microscope also, microns, 10 to the power of minus 6 meters. That much resolution these microscopes will work. Now, in earlier days when the people used to observe under light microscope or compound microscope or electron microscope, they used to draw the pictures of what they have observed under microscope. They used to just draw, that's all. But with advancement of fluorescent microscope, we can observe a photograph of the observed detail. So the fluorescence material is nothing but like film development, you know, in good olden days when cameras were used, films will be developed in labs and all. We used to have photo labs. Right now also we are having some photo labs, isn't it? So the fluorescent microscopes now are working in such a way that the observation can be developed in the form of a film. So that's how the photos of observed things can be brought down. So when you take microorganisms, you'll find microorganisms everywhere. Soil, yes, of course. Water, yes. Air, then plants, animals, including humans, isn't it? So everywhere you will find microorganisms. Even on waste, even on the waste material also you will find microorganisms. So the total environment where you find microorganisms is omnipresent. So remember this word, omnipresent. I hope you know omnivorous word.
Yes, ma'am.
Yes, somebody say.
Ma'am, may I?
Yeah, go ahead.
Ma'am, omnivorous animals are those organisms which eat both plants and animals too.
Yeah, omnivorous we designate to the animals which are both herbivorous and carnivorous, isn't it? They can go for both veg and non-veg. That's called omnivorous. Human beings are the best example of omnivorous. That's all. Now here omnipresent. Omnipresent means the microorganisms are omnipresent. So omnipresent means they are present everywhere. Now when we go for the different types of microorganisms, we talk about the conditions where the microorganisms can survive. If they are present everywhere, how they are surviving everywhere? That's the point of interest over here. Now here comes few new terms. Okay, microorganisms generally will grow under favorable conditions. We use the word favorable. What does mean by favorable conditions for microorganisms? For example, adaptations. Yes, of course they have adaptations. We'll see in detail when we get in about each and every organisms. Area. Here we are trying to give a general concept regarding all microorganisms, whether it may be a fungi, a bacteria, a virus, algae, or some other protist. So all the organisms we are calling microorganisms, and I'm giving a general account. Okay. Yes, Arya. So when you take the favorable conditions in which the microorganisms will go, first we'll talk about temperature. So the temperature usually will be 25 degrees centigrade to 40 degrees centigrade is called favorable condition for microorganism. First thing. Then what else they need? They need the medium in which they will grow. Okay, medium is the thing we call as organic material, like the food material in other sense. Okay, the organic material, if it is sufficient, the microorganisms will grow. Yes, kind of minerals. Yes, Veda, kind of minerals, organic material. We are talking about favorable conditions where numerous microorganisms will divide and grow. Okay, then that's called the medium. Okay, then after medium, we are having different conditions called humidity. Okay, the humidity levels in which they are to grow and divide is also another favorable condition. Along with temperature, medium and humidity, several other factors also plays role in growth and division of microorganisms. It depends. Yeah, Akshata, that's why I gave the term favorable condition. If they go for unfavorable condition also, they can grow. Okay, how they can grow, that I'll explain with examples. Okay. No, Akshata, some microorganisms will not get killed. They'll grow actually. Okay. Arya, humidity has to be moderate. Yes, it shouldn't be too low and it shouldn't be too high. Favorable conditions we are talking now. Okay. So several other factors also plays a role in growth and development of microorganisms. Now, when you take the several other factors, the first foremost factor is oxygen. Presence or absence of oxygen. Based on that, we call aerobic microorganisms and anaerobic microorganisms. I hope you know these terms. No need to explain, isn't it? Aerobic presence of oxygen, anaerobic absence of oxygen. So some microorganisms are there which can grow even in the absence of oxygen also. Okay. Then, other than oxygen, several other factors I am discussing which are favorable. Only favorable conditions, and in general I am discussing. It includes virus, bacteria, fungi, algae, everything. Okay. Other than oxygen, what other factor which will help the microorganism to grow is so here turbidity means, suppose if you take a medium, assume that this is a sugar, assume you have taken on a plate a sugar. Just assume there is lot much sugar without any space, filled up sugar in a plate. Now, if you add some water, some of the sugar gets dissolved and some part of the sugar may not get dissolved because we have taken lot much sugar. You need stirring, isn't it, to get dissolved all the sugar. I'm giving a crude example to make you understand this word. We have taken a plate in which you have taken full of sugar. and you added few drops of water, only part of sugar gets dissolved immediately and remaining part couldn't able to dissolve until unless you go for stirring of the sugar. Okay. Now, this example we can compare with turbidity. Turbidity is the point of medium where some material is available for the organism to grow and some material is not available to grow. In this example, the sugar which is dissolved in water is available for the microorganism to grow, and the sugar which is clumsy and not mixed with water is not available for the growth. So that's called turbidity. The turbidity of the medium is nothing but the organic material which is not useful for the microorganism to grow. So that is another factor which determines the physical condition of microorganisms. So these are various factors which will help microorganisms to grow fast. Remember the word fast. Grow fast means what? They divide and redivide continuously.